Okay, good afternoon, everybody. I hope you can hear me. I've just got to use the computer microphone because my headset died. But this afternoon, we're going to complete our discussion about cell structure, finish off talking about the prokaryotes, and then very briefly discuss the structure of eukaryote cells. Okay, so to complete our discussion about the prokaryote cell, one of the features, really important features, especially in determining clinical significance of prokaryotes, is whether or not they are motile. And today you're going to learn that there's a couple of different ways in which prokaryote cells can be motile. The most common, the most frequent is that they make use of flagella. These are long proteinaceous extensions from the cell, and they enable the cell to be motile, to swim through the medium. Many bacteria, in fact, as long as the surface is damp, can even swim over a solid surface, and that we call swarming. That's done by the same mechanism. So flagella are very long extensions, very, very difficult to see, though, under an ordinary microscope because they are so thin. So we usually have to use special staining techniques in order to visualize the flagella. And what kind of distribution the flagella have is often an important clinical consideration in deciding what an organism actually is. So this is called Peritrichous Flagellation. You can see here, I'm just moving some things out of the way that you can't see. Here's the bacterial cell here. These are visualized with electron microscopy. There are staining techniques which will make flagella visible under light microscopy as well. But in fact, by far the easiest way of visualizing them is to look at it under electron microscopy. So here's the cell here. Each of these threads is a flagellum, and you can see they adopt this lazy S shape in space when we prepare them. And I'll refer to that in a second. Peritrichous flagellation is when the flagella emerge from all over the cell, all around. This here is called monotrichous. Trichous means hair. So this is monotrichous, one hair. And here is the flagellum here, again, adopting this lazy S shape. And this is called Lophotricus. Lophotricus actually means in a bunch. So here we have several flagella all emerging from one spot, and these are emerging. These are polar Lophotricus flagella in a bunch at one end of the cell. These here are referred to as being amphitrichous. They are at both ends. This is actually this whole thing here is a spirillum. It's a spirilla bacterium, but they have a bunch of flagella at one end here and a bunch at that end there. And those are amphitrichous and polar. So there's Lophotricus, one bunch, polar at the end. These amphitrichous at both ends and in a bunch. So the bacterial flagellum is a very interesting little structure. It's in fact really, really complex. When we look at these flagella here, you might be tempted to think that the flagellum moves like this, right? There are waves that pass down the flagellum to propel the cell through the medium. That's not how it works. In eukaryotes, that is how the flagella work. But in prokaryotes, that is not how they work at all. Instead, this shape, this here, is an artifact of us taking the flagellum and pressing it flat. In actual fact, the prokaryote flagellum in space is a spiral like that through space. And it doesn't beat. In fact, it spins like a propeller. It spins in the medium. So the way in which that's done is that it actually has a little motor, a real little motor that is embedded in the cell membrane. There, here you can see here, this is for a gram-positive. Many gram-negative bacteria also have flagella. It's a little, the way it's embedded is a little bit more complex because they have the two membranes. They have cell membrane and the outer membrane. This is a bit easier to understand. It's gram-positive, so there's just the cell membrane. These discs here are proteins. And this whole system uses ATP, and the ATP actually drives this, these discs around, and the flagellum spins like a propeller, like a screw-shaped propeller, and the cell moves. Now, there is something a bit strange about most prokaryotes, and that is that they can only move in one direction. I know it sounds strange, but it is true. When the cell is moved forward, all of the flagella, depending on how many they have and how they're distributed, they are organized so that they beat in a coordinated fashion to propel the cell. If the cell wants to stop moving, it simply reverses the direction of the little motor. They still spin, but they fly out and they spin in a disorganized fashion. And the cell actually tumbles. So there are two, we recognize when prokaryotes are moving, we recognize these two motions, forward and tumble. So forward, tumble, tumble. Here's the thing. If the cell is attempting, let's just say here on this side of the screen here is something attractive, okay? And our cell is over here, this side, and it wants to move. It has to move in that direction to get to the nutrients or whatever. The cell has a way of measuring an advantage over in this direction here. So let's just say it starts its flagella beating in an organized fashion, but now instead of, I've got to get this organized now with my finger. Instead of just moving straight across, right, which is unlikely, it's going to move in this direction. Let's just say it tries to move. It moves to more or less in this, up to here, right? So it's going to move, and when it starts to move a little bit up here, it starts to get further away from the attractor. So as soon as that happens, all the flagella, it's the reverse, and it tumbles. Now it tumbles until such time as it picks up the signal again. And now it starts this direction. Now it's getting further away again, tumble, and instead it moves up in this direction. So it does this in fits and starts, moves towards, as it starts to lose the signal, it starts tumbling until it picks up the signal again, swims until it loses the signal, picks it up. And so what it does is it moves across in a set of steps. It doesn't move if it was here and it wanted to be here. It doesn't go straight across. Instead, it goes in a set of steps, gradually making its way until it finds the place where it wants to be. It's inefficient, it's true, but it's the only way because the cells cannot reverse. They can only go in one direction. They can't reverse. They go in one direction, tumble, find the signal again, travel a bit until they lose the signal, tumble until they find it again. There's only one set of bacteria which can actually move forward and back, and those are the spirochetes. Refer back to see what a spirochete is. They're the one group of prokaryotes which can actually move back and forwards. The others, those with ordinary flagella, cannot. So this whole system here is driven by ATP, and it's a very complex and very energy-rich system. It uses a lot of energy, but very, very advantageous for the prokaryotes that do have flagella structures. And bacteria will, in fact, grow flagella and lose them at different times as well. Sometimes they will only grow flagella under particular circumstances. And there's a whole branch of engineering called Nano Engineering, engineering on a microscopic level, who are very, very interested in this little motor here. Because this is a real motor. It's run in actual fact by electrical energy, by the energy of electrons imparted to it by ATP. Okay, so here another set of extensions from the cell. These are, the first set are relatively short. Here, you can see here, these relatively short hair look like hairs extending from the cell. And these are called the Fimbriae. And they are, their usual function is to actually attach bacteria, to stick them to the surface. And they may be very important in establishing infection and also in establishing microfilms on surfaces. Those are the Fimbriae. And there is another set, very similar structure to the Fimbriae, but which are much longer. And these are the Pylae there. And the Pylae, there are various functions to them, but in certain bacteria, one of their functions is to impart another kind of motility to bacteria. Some bacteria, especially cyanobacteria, can actually glide across a surface. And they can move in a slow, smooth movement across a surface. And that's called gliding motility. There are other bacteria which when you watch them, they actually twitch like this and move in a particular direction. That is called twitching motility. Both of those kinds are imparted by the activity of these long extensions called Pylae. There is another kind of pilus. The singular is pilus, the plural is Pylae. There is another kind of pilus which has a completely different function, and these are called the sex Pylae. Here is a cell here which has extended a sex pilus, and it has extended it to another cell here. This cell here, this one is called a donor cell, and it has a plasmid, one of the small circles of DNA, with the information on it, which enables this cell to create a pilus, sex pilus, extend it to a recipient cell that lacks that plasmid. It builds this pilus and it pulls up close to this other cell, and it makes a copy of the entire plasmid, and it donates that copy to the cell. On that plasmid, in addition to the information for building the sex pilus, there are often other pieces of information, especially resistance information or that sort of thing, as well as the information which will turn this cell now into a donor cell. So this is a way of exchanging genetic information. It's a one-way exchange, but it's a way of exchanging genetic information. And it is that, in a way, is sexual reproduction in a way, because that is what sexual reproduction entails. That's the really important part of sexual reproduction, is the exchange of genetic information between individuals. It's not true sexual reproduction, but remember that they are in fact exchanging genetic information, and this is called a sex pilus between the two. It establishes the contact, then it pulls the cells close together, and there's a one-way transfer of the genetic information from that plasmid. Okay, so for the moment, that's all we're going to talk about about cell structure in the prokaryotes. Now let's have a look at the eukaryotes. I'm going to discuss this I'm going to discuss several different aspects of the eukaryote cell, But again, this is something of a survey. The structure of eukaryote cells is in fact extremely complex, and their functioning is much more complex than that of prokaryote cells. The reason being, of course, they don't only possess a membrane around the cytoplasm, a plasma membrane around the cytoplasm. They also possess membranes which enclose other areas of the cytoplasm. But those membranes perform exactly the same function in a way as that plasma membrane around the cytoplasm, because what they allow is for conditions inside on one side of the membranes to be completely different to the conditions in the cytoplasm. They allow the cell, the eukaryote cell, to concentrate function, to isolate function to particular areas. It makes it very, very efficient. And these different areas, these different organelles inside the cytoplasm communicate with one another extensively. The activity of all of the organelles is coordinated, and they exchange information through the cytoplasm. Okay, so let's have a look first of all, basic kind of eukaryote structures. There we recognize two principal kinds of eukaryote cell, and they are the cells which belong to the whole group of the plants, and then to that whole group, the animals. And there are some singular differences between the two kinds of cell. They have enjoyed a lengthy separation in evolutionary terms and show some marked similarities. They share a common origin originally, but there are considerable differences as well. So I'll discuss the animal cell first of all, just the briefest of characteristics. We'll discuss the different organelles in more detail in a second. First of all, of course, the cell is defined by its membrane around the outside, the cell membrane or plasma membrane, whichever you. But in the animal cells, this cell membrane provides a way of communicating directly with its environment, in that the cell can actually take in parts of the outside. It can actually swallow, if you want to think of it that way, but it can also excrete into the environment, and secrete or excrete. It can excrete waste products and things like that. It can secrete products which it has actually synthesized. So in that way, this eukaryote cell immediately differs from the prokaryote cells, because prokaryote cells were completely unable to do that. They cannot take in stuff from macromolecules and particles and things like that from the environment. Instead, they had to release enzymes and then absorb the products of extracellular digestion, absorb them through the cell membrane. The animal cells have a much more direct way of being able to take in materials. So first of all, both animal and plant cells, the eukaryote cell tends to be very much larger than prokaryote cells. And some eukaryote cells are large enough, in fact, to just be visible to the naked eye. So the first thing to notice about the animal cell, apart from what I've already told you, is that there is no cell wall around the animal cell. This cell here is being shown as if it was isolated. But in multicellular animals, by definition, animals are multicellular. But in the multicellular organization, these cells are in conjunction with all sorts of other cells to make up a tissue. And the cell membranes will actually be tied together with protein bonding to give the structure and to the tissue. Um, I'll just point out a couple of the really important organelles, but we'll talk about their function in a minute. The first thing to realize, of course, in common with the prokaryotes, the cytoplasm is absolutely full of ribosomes. And that is a reflection of the fact that all of these cells obey the central dogma, which is the DNA is transcribed to messenger RNA, which is translated to protein by the ribosomes. Ribosomes occur free inside the cytoplasm, but they also occur bound to another set of membranes inside the cell called the Endoplasmic Reticulum, which we'll discuss in more detail in a minute. Also inside the cytoplasm are many other kinds of organelle, each distinguished by having an outer membrane of some sort and often another set of membranes inside. Their structure tied to their function. There are several kinds that we will discuss. The most important, well, not the most important organelle, but the most prominent organelle is one which is usually easily visualized under the microscope, most easily visualized under the microscope, is that of the nucleus. Now here is the nucleus in the center here. The nucleus, in fact, has a double membrane around it, and it encloses most, not all, but most of the cells, of the eukaryote cells' DNA. There is a little bit of DNA elsewhere in the cell, as I'll discuss in a minute. So also the other thing to notice is there's no cell wall around the animal cell, as I told you. Instead, there's just the cell membrane. Contrast that with the plant cell. Plant cells and prokaryote cells around the outside, around this outside of the cell membrane, they have a cell wall. Animal cells do not. Other organelles are scattered all the way through the cytoplasm. We'll talk about their structure and function in a minute. If we want to contrast this structure of the animal cell to the plant cell, first of all, the first thing you notice is the plant cells have this prominent cell wall around the outside. That cell wall, its primary structure is made of cellulose. That is how it is structured when it is first laid down. And in many plant cells, in the leaf, for example, those organs, you only see the cellulose cell walls. But in the rest of the plant, the cell wall may become extremely elaborate. It may become extremely thickened with other substances. And those other polysaccharides and things, very tough polysaccharides get laid down. That is what makes up wood. Wood is constructed from these substances laid down into the cell walls of the plant. On the inside of that cell wall, the cell itself bears a lot of similarity to the animal cell. There's a cell membrane around the outside, and you recognize many of the same organelles. The cytoplasm contains ribosomes, obviously, all living things do. The ribosome also contains all sorts of other structures. Look here, this is called the Golgi, and there is Golgi there. And here is the nucleus, there's a nucleus, very similar in structure. Here, all around here are these elaborate folds called the Endoplasmic Reticulum, the same in animal cells. So there are many, many similarities. There are a few clear differences. The first is this very prominent feature of most plant cells. This here is called the central vacuole. It's very, very large, and it's filled with basically with water, but maybe some solutes as well. In animal cells, vacuoles also do occur, but they tend to be very small, and they are usually associated with feeding. We usually recognize them as food vacuole. It's a pocket of membrane that's been pulled in bringing in food of some sort. It's then closed off as in animal cells as a vacuole, and various things can happen to it, as we'll talk about in a minute. In plants, this central vacuole here has an entirely different function. And that is that it contains salts, and it, because it does, water tends to flood into it and swell it. And when the central vacuole swells, it swells the whole cell, and it pushes the cell up against the cell wall and creates pressure inside the cell. The cell wall is strong, so the pressure doesn't burst the cell. It presses it up against, and this, especially in the land plants, this is one of the things which gives plants their strength to stand up against gravity. It's one of the things, not the only thing, but it's one of the things. You know this. If you have a plant, a house plant, whatever, and you forget to water it, and it begins to dry out, what is happening inside that plant is that the central vacuoles in the cells start to shrink, and the cell loses pressure. It actually pulls away from the cell wall, and the plant will wilt. It loses its strength to fight against gravity. Water it again, and soon the cells plump up because water floods into the central vacuole, swells it, and swells the cell up against the cell walls again, and the plant will then start to stand upright. So that's one big difference, this fluid-filled central vacuole. There's one other plant cell, animal cell rather, that has vacuoles that we'll talk about in a while. The other major difference between the plant cell and the animal cell, of course, is that plant cells are photosynthetic. And where in the prokaryotes, we heard that the functions of photosynthesis are evolved in prokaryote cells. Those photosynthetic functions in prokaryotes are associated with the membrane, with the plasma membrane around the outside of the cell. But in the plants, instead, they are associated with these specialized organelles called the chloroplasts. That is where the plants get their predominantly green color, because the chloroplasts are full of a molecule called chlorophyll, which is central to the trapping and utilization of sunlight. So the chloroplasts only occur in plant cells. They do not occur in animal cells. Most of the other organelles, in fact, are common to both plant and animal. Mitochondria, for example, endoplasmic reticulum, Golgi, all of those sort of things. These are common features of both animal and plant cells. So the differences, the huge fluid-filled vacuole in plants, not in animals. The cell wall, plants, not animals. Chloroplasts, plants, not animals. Those are the three major differences. Okay, so let's have a look at the structure and the function of some of the organelles in particular. Where I don't clarify the differences, you can assume that similar things pertain to both the plants and to the animals. Our discussion of the nucleus is one of them. So the first thing to note, the nucleus contains most of the eukaryote cell's DNA. In prokaryotes, the DNA, the major part of the DNA, is a large circular chromosome. In the eukaryotes, that is not true. Instead, the eukaryotes have several chromosomes usually, and they are always linear. They are single strands of DNA, very long, which are complexly woven inside the nucleus. There are two states of DNA that I will describe to you. So around the outside of the nucleus, there is a double membrane. It's the two plasma membranes around the outside. And those, the nucleus has to communicate with the rest of the cell. There's one important reason why that you know about already, the central dogma. DNA is transcribed to messenger RNA. Well, that means that messenger RNA is produced from the genes of DNA inside the nucleus. And messenger RNA now needs to be translated to protein. And that takes place inside the cytoplasm. So somehow messenger RNA has got to be synthesized in the nucleus. Then it's got to be exported to the cytoplasm. And it is exported through these pores, the nuclear pores, which penetrate the membrane. These are very complex, and they govern the export of the messenger RNA, but they also govern the importation of materials needed, for example, for synthesizing. You've got to import the enzymes and everything that are associated with the process of transcription. You have to import all the materials to create the messenger RNA. You have to import all the materials to allow for the replication of DNA. DNA has got to be replicated over and over and over again. So stuff needs to be brought into the nucleus so that can happen. All of these importations and exportations take place through these nuclear pores, which are quite complex in their structure. Now, inside this double membrane, all of the DNA is distributed. When we look at the cell as it is here, the DNA is distributed throughout the nucleus. The DNA is wound around specialized proteins called the histone proteins. The DNA has to be extremely well organized. It doesn't just lie there randomly and loose inside the nucleus. It is extremely well organized because the cell has to be able to efficiently access the genes which need to be operated. So that is part of the function of these histone proteins, is organizing the DNA. For the most part of the cell's lifespan, the DNA is in an uncondensed state. It is expanded. It is throughout you can think of it as being pulled out, strung out throughout the nucleus here. And this, when we look at this under the microscope, we see this rough, grainy texture here. It's referred to as chromatin. So that is chromatin. There's one area of the nucleus which is often extremely prominent. And often, if you stain a cell, often this area will stain darkly, or more darkly than the rest of the chromatin DNA. And this is an area called the nucleolus. And the nucleolus has genes on it which are responsible for the production of RNA, but not messenger RNA. There are many kinds of RNA in the cell, and they perform all sorts of different functions. We'll hear about it later on when we talk about DNA, about nucleic acid function in the cell. But I'll give you an example. The ribosomes themselves, we heard before, were constructed of several kinds of ribosomal RNA. It's structural RNA. It does have some enzymatic ability, but that ribosomal RNA, for example, is one of the RNAs which is produced here in the nucleolus, and the cell needs a continual source of this RNA. And so these genes that are responsible for producing it are constantly operating here in the nucleolus. So this is what the nucleus will look like for most of the life of the cell while the cell is doing all of its normal operations, accessing its genes, transcribing them, etc., etc., etc. But when the cell reaches a certain size, then cues are sent to the cell that it's going to need to start to divide. And when that happens, the chromosomes inside the nucleus need to be organized in a slightly different way. Say we refer to it as condensation. These long, thin strands of DNA with their histone, wound around histone proteins, start to get wound up, and they start to contract. They start to contract and contract and contract into thicker and thicker segments until, in actual fact, under the microscope, these chromosomes actually become visible as individual entities. And those processes take place during the two major processes of cell division in the eukaryotes, which are mitosis, the normal divisions of cell growth and everything, and meiosis, which is specifically for sexual reproduction. So during those processes, all of this granular chromatin, DNA, condenses into visible chromosomes. At the same time, in the eukaryote cell, as it begins to divide, the nuclear membrane actually disappears completely. And the chromosomes then are free inside the cytoplasm. That is so that they can be moved around. We'll hear some more reference to that when we talk about cell division and things. But the chromosomes actually become visible inside the cell, and they can be moved by the cell so that they can be moved apart. They're duplicated, for example, and the duplicates are moved apart from one another so that the cell can divide. Once the cell has finished division, the chromosomes unwind so that they can become functional again. The nuclear membranes are reconstructed around them. So now, often closely associated with the nucleus and with the nuclear membranes, there are elaborate folds of envelopes which surround in sheaths all throughout the cytoplasm. And this is a folded transport network. It's called Endoplasmic Reticulum. And there are two kinds of endoplasmic reticulum. Here, first of all, close to the nucleus here, and actually communicating with it because it sends stuff into the nucleus. But all the way around here and throughout the cytoplasm often, there is these folds and envelopes here. Membrane, this is all membrane here. And you'll realize that there's membrane, and then there's a space in the middle. And that is called the lumen. And there are two kinds of endoplasmic reticulum. The first kind is often the most extensive. And here it is, it's called rough endoplasmic reticulum, or RER, rough endoplasmic reticulum. The reason for that is that there are ribosomes stuck to the membrane on the outside. I'll describe why in a second, but just accept for the moment. When you look at the rough ER under electron microscopy, the outside, if you can just make out here, it looks like sandpaper on the outside. There's so many ribosomes stuck to it. That's why it's called rough endoplasmic reticulum. The endoplasmic reticulum is the site of protein synthesis, of much of the protein synthesis in the cell, but it also is a processing plant. There are, once protein is synthesized, it is actually synthesized by the ribosomes on the membrane and is pumped into the lumen of the endoplasmic reticulum. Inside the lumen, all sorts of processes can take place to modify that protein. And once the endoplasmic reticulum is finished with the processing, it will bud off little pockets of membrane, little spheres of membrane containing the processed protein called the vesicles. And those vesicles can actually be moved. And they can be moved to various places in the cell, or they can be moved to the cell membrane itself so that stuff can be excreted. We'll hear about one of the prominent destinations of this in a second. So the rough endoplasmic reticulum is mostly responsible for protein synthesis. So the way in which this works is that messenger RNA, which is sent out from the nucleus into the cytoplasm, as soon as it appears in the cytoplasm, ribosomes will grab onto it. When they grab onto it, there is an address on the messenger RNA. The ribosomes are actually instructed what they need to do. Some of them just stick to the messenger RNA in the cytoplasm, produce their products, and those often are excreted straight away from the cell. Others, there is an address on it which tells the ribosome to attach to the endoplasmic reticulum. The ribosome attaches, and now when it synthesizes its protein, instead of just liberating it into the cytoplasm, it pumps the protein product, into the lumen for processing. So the ribosomes are actually instructed what they should do. These ribosomes that are stuck to this endoplasmic reticulum are there because they are busy synthesizing proteins, which is being pumped into the lumen of the endoplasmic reticulum. So that's the first kind of endoplasmic reticulum, rough endoplasmic reticulum. There is another kind called smooth endoplasmic reticulum. Here it is here. Depending on the type of cell, it is often not as prominent as the rough endoplasmic reticulum. But it is also the site of synthesis, but it is much more closely involved with lipid metabolism, with lipid synthesis and modification. And the smooth endoplasmic reticulum, not as folded, it tends to be more like pipes extending through the cell. It's the site of synthesis of much of the cell's membrane. The cell needs to be producing membrane the whole time. It needs to be producing membrane which can be used to construct organelles, for example. It needs to produce membrane so that the outside of the cell can grow, the cell membrane can grow. All of these, the cell needs a constant supply of an actual membrane, a phospholipid membrane. Remember, that's a lipid, phospholipid. So it's here that phospholipid is synthesized and organized. In addition, the smooth endoplasmic reticulum is the site of synthesis of steroid hormones, for example, which are excreted from the cell. And the other thing about the smooth endoplasmic reticulum is it's the site of detoxification. All cells have to deal with toxins. We take in toxins the whole time, mildly poisonous, some very poisonous in very low quantities, etc., etc. And it's in the smooth endoplasmic reticulum the detoxification takes place. So, for example, in us, our liver is one of the sites of major detoxification activity. And so our liver cells are absolutely packed with smooth endoplasmic reticulum. That's just one of the things, of course, which they detoxify, alcohol. That's one of the principal responsibilities of smooth endoplasmic reticulum in the liver. Okay, so I told you that this rough endoplasmic reticulum would process proteins and then package them into vesicles for communication with other places, either with the outside of the cell, but often with other places inside the cell. The vesicles can actually be moved. And one of the important sites that they go to is this extraordinary organelle called the Golgi Complex, or the Golgi apparatus. And the Golgi is a set of envelopes lying close to one another, which we could think of in a way, an analogy would be that these are a set of factories which are each producing a product and then communicating with another factory. They're communicating with another factory. Each stage is performing another refinement. So the story will begin here at the endoplasmic reticulum. And the endoplasmic reticulum, an early version of a protein may be produced. Now, the thing to understand is that proteins very often, in order to be really, to achieve their proper activity, they need to be extensively modified. They may be modified to a certain extent inside the endoplasmic reticulum, but then that product is exported to the Golgi apparatus. And it's exported in the form of a vesicle, a transport vesicle. So here comes a transport vesicle containing the rough draft of a protein. It binds to the Golgi, and some sort of modification takes place here inside the Golgi. The Golgi then manufactures vesicles of its own and sends them up somewhere else in the Golgi. Usually they make their way through the Golgi step by step by step. When the protein is finally finished, when it's achieved its mature form, then a vesicle is budded off. A secretory vesicle is budded off from the Golgi containing the final product, and that is sent to its final destination, to the outside if it needs to be secreted from the cell, which is often the case, but to some other destination, another organelle maybe, or something like that. Again, the protein is addressed. It actually has an address on it, which instructs the cell where it needs to go. Many of the products, by the way, are secreted from the cell, as well as being distributed throughout the cell. Okay, so here is another, remember the Golgi is common between plant and animal cells. It is organized a little bit differently in the plant cell. But the function is essentially the same. Here is another organism, another organelle shared between plant and animal. And these are the mitochondria. The mitochondria are the site of synthesis of adenosine triphosphate, ATP. And that is, we always refer to this as the biologically useful energy source. That means that this is an energy source which is very easy for metabolic processes to access and use. It's not a long-term product, however. It's very short-lived. So it needs to be continually produced. And it is produced by the processes of cellular respiration. Cellular respiration is the process whereby the cell takes a fuel, sugar or a lipid or something like that, and actually effectively burns it with oxygen to produce energy, a chemical energy, which can be bound to ATP for use by metabolic processes. We will talk about this process extensively, okay, because this is one of the most important metabolic processes that take place in both prokaryote and eukaryote cells, and it's essentially the same process in both. The mitochondria are complex little organelles. They also have a double membrane. So there is a membrane around the outside, there called the outer membrane, and there is an inner membrane which is extensively folded like this. That is so that it has a lot of surface area, because all of the substances, all of the enzymes and everything that are needed to perform cellular respiration are embedded in this membrane all the way around. So it needs a lot of surface area to get that done. And again, the function of these membranes is to regulate conditions on the outside and on the inside. So the outer membrane here creates an interior environment for the mitochondria. This inner membrane here divides that inner environment into two parts. There's an inside here, and there's an outside, which is between the outer membrane and this inner membrane. These folds here are called the cristae. And the equivalent of the cytoplasm in the mitochondria is called the matrix. So all of the processes of cellular respiration depend upon being able to maintain a difference in conditions on one side of the membrane and on the other, as we will hear later on. Now, there are a couple of things which to take note about these mitochondria. First of all, look their size. Look at the size, about 2 microns or so, about the size of a prokaryote, of a bacterial cell. Second thing is, these mitochondria have their own DNA. Now, you remember I've emphasized, every time I talked about the nucleus, I said it contains most of the cell's DNA. Not all, but most of the cell's DNA. Here's one other place where DNA occurs, and it is called mitochondrial DNA. It's a small amount of DNA, and it is in the form of a circular chromosome. Where else did we hear about circular chromosomes, single circular chromosomes? In the prokaryotes. Not only that, the mitochondrion also possesses its own ribosomes, and it can produce some of its own proteins, not all of its proteins. Some of its proteins can be produced. The ability to produce all of them depends on stuff being imported from the cytoplasm as well. But it can produce some of its own proteins from its own chromosome. Bear that in mind, okay? You will hear mitochondrial DNA referred to frequently if you pay any attention to forensic science. Any of these studies that look at DNA sequencing for identification purposes will often refer to mitochondrial DNA. Because mitochondrial DNA tends to be, it's very well protected inside the mitochondria. And the molecule is quite small, so it tends to be preserved a lot better than the rest of the nuclear DNA. So it can be used. One of the interesting things about our mitochondria, however, is that we all inherit our mitochondria from our mothers only. So we do not inherit mitochondria from our fathers, only from the mothers. But the egg contains the mitochondria, which will reproduce to become the mitochondria of any resulting fetus. So if you use the mitochondrial DNA for identification purposes, it takes you through the maternal line. So for example, whether you're male or female, you have your mother's mitochondria. Your mother inherited her mitochondria, which are identical to yours, from her mother, and she from her mother. And so you carry mitochondria which are identical to your entire maternal line, all the female matrilineal line, all the females in your family share mitochondria with you. So do your siblings. Okay, so those are the mitochondria. Very, very interesting. Tremendously important. They tend to be abundant in cells which require a lot of energy, and they are particularly active when there is a demand for ATP. Okay, so the next thing to describe this much more common, I should say, in animal cells than in plant cells are these organelles called the lysosomes. And the reason for that is that the lysosomes are a particular type of vesicle which is formed in the Golgi complex and contains digestive enzymes. So what happens in animal cells is that animals will take in material from the outside, from their environment, either containing particles or whatever, but they'll effectively swallow, producing a small vacuole, which can be moved through the cytoplasm. And it's those vacuoles containing stuff that needs to be digested are moved close to these vesicles called the lysosomes, and they unite with the lysosomes so that the digestive enzymes are mixed with the foodstuffs, digest everything, and that can then be absorbed by the cell. The reason for this, the whole elaborate structure, and the reason that these are produced in the Golgi is that the cell needs to be protected from its own digestive enzymes. You can't have these powerful enzymes just drifting around inside the cytoplasm, because many of them, for example, are will break up proteins. They will break up protein. They will destroy membrane. All these sorts of things. The lysosome is specially designed to be protective of these digestive enzymes, to stop them destroying essential elements of the cytoplasm. Vacuoles, I've told you about the plant central vacuole, which is a very large vacuole containing a solution of salts. But there are other vacuoles inside the cell, especially in animal cells, smaller vacuoles, and for instance, to bring in the food that I've mentioned already. But they may also have some other functions. They may sometimes be used for storage. So, for example, here in animal cells, here each of these here is an individual cell. And these I mentioned in another class. These are the adipocytes. These are the sites of storage of lipid and fat. So these are our fat cells. And here you can see this whole area here is a vacuole. It's a single vacuole, and it's filled with this lipid droplet. It's so huge, it's crammed all the cytoplasm to the side of the cell. There's the nucleus here. Thin layer of cytoplasm around the outside of the vacuole, and this whole lipid mass filling the vacuole in the center. So those are in animal cells, those are the adipocytes. It's almost the only, in animal cells, almost the only very large vacuole that you will see. In plant cells, here is the central vacuole here. And remember something, this is a membrane-bound organelle. It's not just a space filled with water in the middle of the cell. It has an active membrane around it, and that plasma membrane around the outside of the vacuole is responsible for regulating what goes in and what goes out of the vacuole. Again, the central vacuole swells with water coming in from osmosis and presses the cell up against the sides of the cell wall around the outside. Okay. I'm just going to mention also at this stage, because I haven't mentioned it before, you'll realize that the structure of the cell is by no means random. There is a very particular structure for each kind of cell. It has its own architecture, and much of that architecture is imparted by protein itself, which is built into a cytoskeleton. Literally what it says, a skeleton for the cell, comprised of lengthy filaments and tubes of protein. These are active. They can be broken down and rebuilt continuously so that the cell can change shape, for example, or even the cell can move by doing so. But the cell shape is primarily determined by these long strands of protein which fill the cytoplasm and provide the cell with a skeleton. Okay, so here is the distinguishing organelle for the plants, which is the chloroplast. And the chloroplasts are the site of photosynthesis. and are the primary site where plants are able to convert light energy into chemical potential energy. And this takes place on an extensive membrane-bound set of structures inside the chloroplast. Again, this becomes a familiar story. This organelle has two membranes. It has an outer membrane here around the outside, and then an inner set of membranes which are extensively folded. There is a huge surface area here, and they are folded in a particular organization. You will see that some of them are stacked envelopes formed from the membrane, from this inner membrane. These are called the thylakoids here, and thylakoids pile up one on top of the other to form this unit here, which is called the granum. So thylakoids, individual envelope sheaths there, then piled up, they form the granum. This area in between here is called the intergranum. So that when we look at an electron micrograph of a chloroplast, this is what we see. We've cut across now. And what you see here are here's the granum here, comprised of individual thylakoids piled on top of one another. Then these looser intergrana here. So again, what we've got, first of all, we've got the outer membrane creating an interior environment for the chloroplast. Then on the inside here, the thylakoid is an envelope, like that. It has a space in the middle. So that interior space is different in constitution to the outside. And the whole process of photosynthesis depends on maintaining that difference, as we'll see when we consider photosynthesis. So in quite a complex structure. The green color of chlorophyll is embedded in these membranes and gives the plant a fundamental green color, although plants can have other pigments which color them. But the really important active photosynthetic pigment is chlorophyll, and that is embedded in these membranes. Apart from that, the chloroplast also possesses its own circular chromosome. Again, it's beginning to sound like a broken record. Its own circular chromosome. And it also contains its own ribosomes. And much like the mitochondria, it can produce some, not all, but some of its own proteins. So that begins to sound quite familiar. Both mitochondria and chloroplasts share that, although, of course, the mitochondria occur in both animal and plant. Chloroplasts only occur in plant. There's a couple of the other little organelles that you will see. First, very small little vesicles called the peroxisomes. The processes of photosynthesis and respiration depend on the cell's ability effectively to move protons and electrons through particular processes. That is a very dangerous thing to do, because these, both protons and electrons, are highly active. They contain a tremendous amount of energy, and they can do some very damaging things inside the cell. And one of the difficulties is that the cell continually produces very strange versions of oxygen. Oxygen itself is very dangerous. It's a very dangerous element. Reacts with many biological molecules and destroys them. But the cell also produces different kinds of oxygen species, O3 and all sorts of things, which are also very, very damaging. And one of the common products of these processes is this substance here, H2O2, which is peroxide, hydrogen peroxide. And hydrogen peroxide is very damaging. and it needs to be dealt with very quickly. And cells have various ways of dealing with it. But one of them is that cells have these little organelles which are dedicated. One of their functions is a dedicated function to destroying hydrogen peroxide. And these are called the peroxisomes. But the peroxisomes, this used to be thought to be their only function. It turns out that the peroxisomes actually have numerous functions. And one of them is to break down the fatty acids. You'll remember when we talked about phospholipids, we said those tails, the phospholipids, those are fatty acids, the long hydrocarbons. And those are fatty acids. One of the things the cell sometimes needs to do is to break those down, digest them, and metabolize them. And that is the responsibility of the peroxisomes. Here is another organelle. Now this one, these are the centrosomes. And this is one centrosome here. Each centrosome consists of 2 centrioles, one there, one there. And they're a cluster of little microtubules made of protein. They don't look like much, but they have the function that they manufacture all sorts of threads and protein tubes and things that are involved, especially in cell division. Now, the one thing I must tell you is these are particularly well organized in animal cells. They are very difficult to see in plant cells. So there's some difference there, but very well organized in plant cells, in animal cells rather, where you can easily visualize 2 centrosomes. So each centrosome consisting of 2 centrioles. So these are what form a spindle in the dividing cell, and they actively are responsible for the movement of chromosomes. So have a look here. This is a dividing cell. The cell membrane has disappeared. I mean, sorry, the nuclear membrane has disappeared. The chromosomes have formed. You can see them here. They are stained blue here. There. Here are the centrosomes there and there on either side. And they have formed this spindle. All of these are tubules made of protein, which have arisen from here. So this is, these are organizing centers for the production of this spindle of fibers here. And these fibers contact the chromosomes and allow them to be moved inside the cell, moved from one side to the other. Okay, so I've mentioned twice now, in the case of mitochondria and in the case of chloroplasts, these very strange facts that these organelles rather, possess their own chromosome. Their chromosome is circular. They have their own ribosomes, and they make some of their own proteins. All of those things make them sound very much like a prokaryote cell, right? Because prokaryotes also have a circular chromosome. And in fact, when we look at the ribosomes inside the chloroplasts and inside the mitochondria, they look more like prokaryote ribosomes than eukaryote ribosomes. And this has given rise to this theory that the eukaryote cell arose originally from a primitive cell, which actually took in bacteria, which had different functions. So, for example, this here is an early cell, a very, very early cell, very primitive cell, which is going to give rise to 2 principal groups, to the bacteria and to the archaea. We believe that that the process I'm going to describe took place actually in an archaeal cell, not in there is a foundational eukaryote cell, a primitive foundational ancestral eukaryote cell, which was probably archaeal, or more similar to the archaea than to the prokaryotes. But this cell actually took in bacteria. One lot of bacteria were oxidated bacteria. They were bacteria which could use oxygen, which could use up oxygen to produce ATP. And up until that time, these cells were stuck with primitive systems, more primitive systems, which were very inefficient sources of ATP. But then they took in these bacteria that could do this using the process of cellular respiration. And that was a huge advantage. Secondarily, another group took in a group of prokaryotes, very like cyanobacteria, which were able to photosynthesize. And the prokaryotes that did oxidative processes, cellular respiration, ended up forming mitochondria. The ones that could do photosynthesis ended up forming chloroplasts. This line of cells then went on to produce the algae and the plants. This line of cells here, which didn't have chloroplasts, went on to form the animal cells. So that is the theory of how the eukaryote cell actually arose during the processes of evolution. And this is referred to as the endosymbiotic theory. The endosymbiotic theory, a primitive cell took in other cells. Initially, these cells here were commensal there was just an easy partnership, but they slowly became entirely dependent on one another. You cannot take chloroplasts or mitochondria, separate them from their host cell, and have them reproduce and behave as if they were independent. The two are this is now a single unit, the eukaryotic cell. And I think that is it for today. You get a present because we finish a little bit early. Okay, so remember, I will see you on Wednesday, live class. I apologize, I don't know what happened to Zoom last time, but I have set up a new Zoom room, and I will communicate that room to you for you to log in for the live class on Wednesday, 4:05. Okay.